request for a review sheet, so I'll show you what I did for that. And I'll have hard copies of it on Monday as well. So I had down in the weekend stuff, final exam review questions, that's the exam file. So I just moved it up here into the current content and relabeled it as midterm review. But as you'll see, series of questions by category, right? From the first week on cells, then roots, then stems, then meat, the flowers, and fruits. And that's the end of the midterm related material. This is photosynthesis and respiration and hormones, etc. That will be on the final exam. So don't forget about that now. We'll hear about it after the fact. But this includes all the things we have covered and then some. So it gives you a sense of, do you understand that concept? Can you answer that question? And of course, just like on these quizzes, right, the homework indicates something that's important. The PowerPoint slides indicate something that's important. And now 2 quizzes worth of material that point you toward how I'm getting at things, how I'm asking things, etc. So between today and Monday, make sure you get those questions answered about the quiz, about midterm prep. And then Wednesday will be just the whole time to take the test. So no content beforehand, and then when you're done, that's the end of the day Wednesday. Questions before we get going? Let's take a look then. Say overall, results were better than the first one. I think people get the build up, getting maybe the style of questions, what to look out for, etc. And there will be more on the midterm of the shorter answer types, true and false, multiple choice, and then continuing to be a chunk of short answer, drawing, labeling, indicating you understand some bigger process, but more of all those things. So let's start at the top. Most monocots have no secondary growth. That is true, right? Directly off. Taking you back to the stems material now. About what they are, we'll mix them up, and then we got right into the monocots. First thing first, go back to the cambium. Only dicots have secondary growth, right? So I'm getting directly at that. Number 2, dicot stems have vascular cambium, like I just mentioned. Right? Important to notice, in contrast to the monocots that have those intercalary meristems dispersed throughout rather than neatly bundled in some pattern, right? Circular type pattern around the bit. Number 3, this one was a little bit tricky, requiring you to both understand and read closely. The vascular cambium produces xylem to the outside and phloem to the inside. That's false, it's the opposite. Right? Xylem to the inside, always, that's what makes up the interior heartwood of the tree, which gets directly at the next question. Then your secondary xylem is wood, right? And that's true. So I stacked things up, right? So those are connected, directly connected to this one about mature woody stems versus herbaceous stems. And then this shows up on the next page in 13 about hardwood versus sapwood. So there's a lot of interconnection here, opportunity to borrow epidermal growth, and not thorns that show vascular tissue as stems. Three primary functions of leaves, right, that came directly out of the first slide on leaves. That would be photosynthesis, transpiration, respiration. And number 7, what differentiates woody stems from herbaceous stems? Right, at least 2 things. Well, what's the biggest one? That's correct. I would consider that the second one. There's one that's implied in the question.
Herbaceous stems like the grounds.
Right, so the woodiness of the other ones, right? That comes from the secondary growth that is that wood, right? That is that secondary xylem deposition. Yeah, so that's primary. And then correct also, the cork-cambium production of cork and bark to the outside is another distinction with those trees versus herbaceous stems. Number 8, got at that picture I showed, giant sequoia with the hole in the middle. We talked about how this thing survives. And many correctly understood that it can still move water in the xylem, right, around that outside ring. You remember? We're looking at the tree cookie view. That vascular cambium that's producing xylem flow is close to the outside, and right, each year it's making that xylem to the inside, and that flowing to the outside there. So as long as that's intact, right, then growth continues to be possible. And of course, in this example, like part of this is right removed for the convenience of driving through somehow. But as long as that's intact in certain spots, then we can continue. And so here, I was looking for a little bit more detail that distinguishes this tree, right, from other plants. So if I wrote heart/sap on there, that's because I was looking for you to mention that it's the functional sapwood on the outside that's still right, living and transporting water, doing that task, versus the dead heartwood, which is the majority of what was removed, right, while that structural role is gone, the functional transportation role is still served on the outside here in the sapwood. Full points for specificity there. And distinction between bulbs and corms. And again, this came right out of the presentation material on stems. Here at the end, right, they're both stems. One has primarily fleshy leaf with a little bit of stem, one has mostly stem. Right, that just tells us something about its capacity for reproduction later, its ability to survive. So, not a lot of points for that one, but it does keep you thinking about the direct relationship between molecular material and the question. So if there's questions about these that I don't approve, please. Number 10, what does the opening and closing of stomata accomplish? 2 things.
Respiration and transpiration.
Yeah, so water out in transpiration, and then in respiration, it's also actually CO2 out, and another one of those could be CO2 in through photosynthesis. Right? So there's an in roll and an out roll. Sometimes it's different gases, but always water moving out, absolutely, in the stomata. So, very important part of plant function is the relationship between the inside portion of the plant and the exterior environment, and that's the threshold where it's moderated, those garden cells opening and closing. And so I'm going to ask you more about that in the midterm, so make sure if the stomata-related questions here, if you lost points points there or didn't understand, that you give a little more focus there, because it's the foundation for the next stages of talking about in detail photosynthesis and respiration. So by the midterm next week, we'll have talked about all the plant organ systems, right, and put them together into a functioning organism. And then in the second half of the class, then we're going to talk about what those processes are that actually make it work and how it responds to the environment. And like I said just a minute ago, this is the threshold for that exchange. So really, really important to understand what's going on there. Where are most of these usually found is the bottom of the leaf or the underside of the leaf. So knowing the location is one thing, but why are they usually there?
Take it from the sun. Take it from the sun. That's water, so you lose less water.
So you lose less water, right? They're not in direct sun, right? They're a little bit, right? They're covered. It's somewhat of a microclimate under there. It's less windy, right? Less exposure to airflow. So yeah, we can maintain that humid environment on the other side better than we can. And of course, when a plant finds itself in a different set of conditions, we gave the example when I talked about it of aquatic plants, right? If they have leaves on the surface, that's going to be where they want the stomata then for getting oxygen in from the part that's in the atmosphere rather than submerged in water. But by and large, wherever the scenario is most advantageous for managing water loss is where you'll find it. Okay, and 11. How is the seasonal change in the water needs of a flowering dogwood tree reflected in its xylem due to development? So again, I was looking for a little specificity here. I gave you the clue of the flowering dogwood, right? It tells you that that's an angiosperm tree. So, right, by nature of flowering, right? So there's something about those angiosperms, in fact, in that big drawing here. If you got some points if you told me that, right, the water need changes, it's higher in the spring and it's less in the late summer or beyond. And you got all the points if you told me what was going on was vessels were produced early to move a lot of water, right, to facilitate that creating a new canopy each season. And later, smaller tracheids were produced until that brings completion, and then the next year, you see that same process again. So all the terms, right, to get full points there. Vessels early, tracheids late, and I want to be clear that those are two different structures, right? Some described it, identified correctly the change and the size differential and the reasoning, but said like, the vessels become tracheids or something. It's just they make one thing and then later they make a different thing, right? So they don't transform. It's not the same thing but smaller, right? It's two different things that function slightly differently. Here, where's my other line? In here, right? So you can see, while similar, there's a structural difference. So it's two different things, right? Number 12, grasses and stolons. Those look like roots. Go pick up the class. That are stems. How can you be sure that what you're looking at is stem tissue? I was very happy most people correctly understood that the answer here is nodes. Right? We have nodes on stems. We talked about tubers, potatoes, where the eyes are nodes, the importance of the meristematic tissue at the node that can produce a shoot, a root, flower stalk, right, etc. So make sure you're addressing the question that I'm asking specifically. Some told me like rhizomes and stolons are both stems, and I said yes, that's what I told you. So, how do you know though? Number 13, the major difference between sapwood and heartwood. Again, you're getting a sense. I'm wanting you to put the pieces together and use the terms that describe what something is. So here, full points said that sapwood is the functional vascular tissue, like living xylem phloem, doing transportation, and the heartwood is now, it's that secondary xylem, right? It's dead cells on the interior that no longer has that transportation function, just a structural function, kind of a shunting away storage, hide things out of sight function, and that's it. So there was a lot of partial points there for pieces of that. Again, here in the drawing, really get you to think about the nodes, what happens there. Very satisfied with the results. Most people correctly identified this, and the final piece that why it's so important about the tissue at the nodes is that it's meristematic, right? Is that at that site, just like at the root tip, just like at the apical meristem, we can produce different plant structures, and we gave that example of adventitious root production at the node, adventitious shoot production from other location. So essential role. And again, to connect you back to the material that I'm drawing from here. Talking about the nodes, tissue, adventitious root formation. Here's what we find there, axillary buds. So again, understanding that lecture material connects directly to the questions that I'm asking. That's the labeling here. Gave you a word bank, so you just needed to fill in. Okay, epidermis on the top, vascular bundle, palisaded spongy mesophyll. This was maybe the sticky spot on the identifying here. Stoma or stomata, remember, is the empty air space, right? It's just the exchange area, and the guard cells are the actual living plant cells that are moderated by that combination of signals for opening or closing. It's another example of how it adds all up. So I introduced guard cells in leaves, right? They connected to vacuoles from the first week, right? The cellular organelles. They're connected to the first and second wave with the movement of water, right, osmotically into the vacuole. They become turgid and open those guard cells effectively. And then I told you that light is one of the signals that opens stomata. And again, the mention of osmosis. And then when we talk about hormones, I'm also going to tell you that one of the major 5 plant hormones, abscisic acid, is the trigger signal when, right, a certain quantity of light is noted by the plant, absorbed by chloroplast and recognized, that signals then the need for guard cells to swell and open. What's that? Abscisic acid. So again, that's not coming up on this or anything, but when we talk about hormones, it's just another example of how then some later information plugs into this, because it all fits together. So there's the signaling for them to open or shut, right? Light tells it, hey, it's warming up. We probably need to get some heat out of here. Water is pulling hard. We need to let some out, or we need to get some CO2 in and get photosynthesis going good. At the same time, then this hormone signals, okay, do that. Start moving a lot of water in, fill the vacuoles, open up, and remember that little process is happening quickly at millions of individual stomata sites throughout the plant. And it's that overall balance that determines how much is coming in and how much is going out. A few more. Number 16, I basically threw out. So everyone got 5 free points there. I changed what previously had been observe a twig and tell me about it to draw a twig and tell me how you know the age. One thing I want to point out, I was thinking when I said draw a twig that it would be like and you would say, okay, 1 year, 2 year, and here is your bud scale scar. And you could have added more detail, right? Some people had an additional scar or a bud, whatever. That's good. Too many people, which means I need to be clear around the question, drew this and said 2-year-old twig.
Doesn't work.
I thought it was great. I like that answer.
And the reasoning there was, what's the name of the structure that tells you this? I think they were just like, well, it's the 2 annual rings that says it's 2 years old. Right? So I'll be more clear. Well, everyone got the full points for this question. As like I said, I threw it out. So I would call this a cross section, right? Sometimes it's called a tree cookie, right? But in the future, I will be more clear so others won't have that same issue. This said, piece of twig. So just consider that, and I will, again, no one lost any points on that. Draw tree branch that has alternately arranged leaves, simple and petiolate. Label these things, veins, margin, apex, right? Just like I promised you, I don't need you to know, like, oh, that's an acuminate tip or whatever. But here's where the tip is, so if we're talking about differences, that's the point. And very satisfied. Just about everyone got almost all the points on that, at least. And if you drew the petiole but didn't expressly point it out, then I circled it and made an arrow to it for you because you also included the things that I was asking for.
Can terminal bud scars be the same as bud scale scars?
Well, in 1 year they are. So if that's your terminal bud, then this year, that terminal bud, right? So now that was a terminal bud from 2 years ago, right? So at some point they were all a terminal bud. But it's possible that a plant, and likely, grows more than one node in a season, right? So, yeah, both fit, you get the same effect, right? Whether it was a terminal bud this year or 6 years ago, you see that little folded up result. I guess the question is more about the weight there. And my bonuses, the well-dried hardwood or softwood was split into triangles. Why? If you took a stab at things, I generally gave some points. This one I was looking for the rays, medullary rays, through the wood and back to our stems. So in all these things, I want you to understand that I'm looking for information, stems, that indicates your understanding, right? I'm trying to evaluate here, which I can't necessarily do in the homework, are you connecting the dots? So like, when I asked about why that splits, a couple of people told me, like, because it's dry now. And it's like, yeah, but not all dry wood splits easily, and there's a reason that's connected to the structure and the function of the plant, right, rather than just the outside circumstances. So So in this case, right, the ray is what is connecting, right, I said, hey, it's that pith and then secondary xylem here, the wood, where we can store some old stuff. That's what the rays facilitate. And they're also connected to that living vascular cambium closer to the outside. And so those, as they dry, right, they leave hollow tubes that reach from almost the outside to almost the inside, and thus making it much easier to split than without them. If it was all solid, it would be a lot more challenging.
Chris, you said that was the flown ray? Is that what you call it?
Medullary rays. So they connect the cambium, vascular cambium here, which is phloem and xylem, the living portion in the sapwood, all right, all the way to that pith, which was like in the first year, right, before there was even additional secondary xylem deposited. So they were developed right away, and then they grew each year, continuing to connect to that living portion. So things can be transported from all the way, right, if needed. Not specific to trees, right? Perennial hardwood. Christopher?
Medullary rays, they connect, you said, phloem and xylem, not just one or the other.
That's right. So that green layer here, it's in fact two cells thick, right? One making phloem and one making xylem. Again, to that earlier question, phloem to the inside. No, xylem to the inside, phloem to the outside. And the reason why it matters that it be connected to the phloem too is that that's often where things that need transporting for shoving away for safekeeping somewhere come from. Like, let's say some environmental contaminant gets in through stomata, lots of leaves in the canopy, and then it's moved down via phloem, and that's the thing that we want to seal off. It needs a way to get there. So we can't move anything from above in the tree via xylem, right? Because the motion is only bottom to top. And the last bonus was what is unique about the stems of cacti? It's a little open-ended. There was opportunity for fewer or more points based on your answer. What's one unique thing about the stems of cacti? Filled with water, absolutely. Big water storage organ. Got points for that. What else? Something about them being green. They photosynthesize, right? That really stands out. We know that across plant species, there's storage opportunities in roots, in stems, in leaves that manifest in different ways in different species. What's really unique here is that the stem is the photosynthetic driver for the plant. I heard something about the leaves being spines and how that was connected to the stem being photosynthetic. So more context, more points. And of course that one came from the end of stems down here. Here. Photosynthetic, platypil, big flat stems with tiny spiny leaves. That was quiz number 2. More questions on that? So, hang on to that, review it. I want to get it back to you right away in the next class, so you have it for the weekend and potential, exam study and preparation. And now you've got both quizzes. You can hang on to all the homework and review over the weekend too, and hand it to me on Monday. Or if you handed it to me today, I will have it back to you on Monday. And then you can use it Monday and Tuesday as you're preparing. So either way is fine. None of them are considered late or anything. And I'm just trying to make sure that you have all the information possible as you prepare. And of course, bring more questions wrapped up flowers, if you remember, I ended with Parthenocarpy, that specific example of plants that do not necessarily require pollination or fertilization, right? Two separate processes, but still get us a fruit. Generally, then we compromise having viable seed for future generation or diversity. And it was time to then talk about those fruits. These now, non-parthenocarpic fruits, things that are actually the result of pollination and fertilization. And we classify them just like we classify leaves, just like we classify the parts of the stem and root. And here, understand the basic relationships. For me, so we call a drupe something that's thin-skinned, has a central stone or pit, and all the stone fruits fit the bill here, right, as a drupe. I'm not going to say, give me two drupes as an example, but what I do care about is that these, so let's say this is a peach, our example here, of a fleshy, simple fruit. And so inside is the seed, right? That's the actual fertilized embryo, encased in a seed coat. Around it is, right, the pericarp, berry-like around, and carp like carpal or pistil, right? Just the body part is the protection and sometimes food availability or attractant for seed dispersal. And these come from single flowers with one ovary. So that's the part that I want you to pay attention here to as I make these distinctions on the next couple of slides. So simple fruit, just like the name implies, it's not too complicated because it's one flower, one ovary. So one pollination event fertilizes that one ovary, and you get one peach.
Does it still have multiple ovules inside the ovary, and it just only allows one to accept pollination, or is there just one ovule?
Different plants have a different answer to that. Some have backup additional ovules that then, once the first one's fertilized, they shut down. They become non-functional and generally join the ectosperm and become part of the food source. Others only have one. And so I'm going to give you you'll see one flower, one ovary. You're going to see one flower, many ovaries. You can see many flowers, many ovaries. And so the structure tends to look different based on which class it's in. But species to species, the way they answer that question changes. So that's a simple fruit. One flower, one ovary. Here's another example of a simple fruit. Single flowers with one ovary. At this time, no central stone or pit, right, which was what characterized the drupe. We call it a berry. Tomatoes, believe it or not, are a berry, and strawberries are not. Again, I'm not going to ask about that distinction, but what matters here is you understand single flower, one ovary. I think this helps answer your question for this example, Mr. Is that there can be several ovules within the ovary like you're asking about, right? And so here you can see, right, all those yellow bits are individual seeds. So each one of those was an ovule. So that one has several, right, within the single ovary. Meanwhile, one seed, right, one pit on the peach means one ovule. So peaches answered your question no, and tomatoes answered your question yes.
So each seed, then it gets a grain of pollen and becomes a seed surrounded by the the flesh. But they were all ovules at one point inside the ovary, and then they become seeds as the physical grows into the flesh.
Yeah, yes, that's right.
So that's
So
what makes it right.
And each one, to your point, right, because each pollination event only creates 2 sperm cells. One of them becomes the pollen tube, the other divides into 2 sperm cells, pollen grains, excuse me. One fertilizes the ovule, the other becomes endosperm. That situation in the tomato is going to repeat for each one of those seeds. Yeah.
What's the name difference between, the one picture before and this picture?
Between the peach and the tomato,
Yeah.
primary difference is 1 seed, many seeds.
I'm trying to wait, what's the name for the 2 different?
They're
One
both,
seed and
they're
many
both
seeds.
simple fruits because they are 1 flower fertilizing 1 ovary.
Would
Yeah.
it be the difference between like drupe or berry,
That's the, yes,
the main difference?
that would be, yeah, no single pit
Okay.
for the berry, right? And the drupe does have that single pit. But yeah, concentrate on, to Christopher's point, that each ovule becomes a seed. And here, that all is happening within a single flower and a single ovary. In contrast, okay, sorry, one more fleshy, simple fruit example. You'll find lots and lots of our, and I'm using all like things that we classify at the grocery store as fruits, right? But when we're talking botanically about a fruit, it's a seed with some kind of pericarp around it, any kind of fleshy something around it, which it's not always soft and juicy and sweet like a peach or a tomato, right? Sometimes it has a different texture, sometimes it doesn't look appealing, etc. But it's encasing that seed for protection and dispersal. So another simple fruit, again, 1 flower, 1 ovary. These we call pomes. Apples and pears, you might know, as pomes. And pomes have 5 seeds, and so 5 ovules within the ovary, and they tend to have an enlarged receptacle. And so this is where the apple is connected. Sorry, this is the flower, right? This is where it's connected. That was the flower that something pollinated on, and then it literally went down the pollen tube, fertilized these ovules inside the ovary that produced the 5 seeds inside, and all the flesh that we're eating is that pericarp around it. Believe?
Is avocado a fleshy fruit thing?
So avocado's got 1 big seed inside, right?
Mhm. So is,
So from what we've seen so
is that a
far,
drupe?
it fits here, right?
Yeah, it's a drupe, right?
So lots of fleshy, simple fruits. Now, we can start to understand the thing by differentiating it from something else. So that's simple. And again, within the simple, 1 flower, 1 ovary. But let's make a note here that the seed number varies. And we saw peach 1, apple 5, tomato many, and not necessarily the same number every time. Aggregate fruit, raspberries, thimbleberries, fantastic example from the rubus family, are 1 flower, so the same setup. And if you've ever grown raspberries or picked raspberries or something, you've seen, right, that little white flower, lots of yellow stamens around. The berry is what shows up right there underneath. Okay? So 1 flower, but many ovaries. So it's a collection of fruits on a single receptacle. And strawberries fit in here as well. So when you're looking at that raspberry, right, each one of those individual sections, like the little bumps, is technically an individual fruit with a seed inside. You put them all together, all these ovaries at the receptacle, then we call that an aggregate fruit, right? We think of the raspberry as one thing. And it did come from one flower, but it fertilized several ovaries to get there. So again, the receptacle, at the point, is, well, I guess to the raspberry example, the best way to talk about it would be, so on a blackberry, it's very similar, right? You got all these individual bits. And for the berry pickers out there, you know, one of the primary differences, when you pick a raspberry, right, you slide it off, it's hollow inside, and that white kind of cylinder stays on the plant, right? And that's the receptacle. So that part that stays, you pick the raspberry, right? You get like a U shape, a berry, and this thing connects back to the plant. That's the receptacle that they're formed around. On the blackberry, the receptacle comes off at picking, so that whole thing, it gets eaten. It's softer. So those are aggregate fruits. And to round out, here we've got the multiple fruits. Many flowers. So remember, simple had one, aggregate had one. Here we have, it's variable, but it's more than one. Many flowers, and each one then has at least one ovary. Call that then many ovaries. I'm sticking with my food examples. Pineapples are a great one. So pineapples are an interesting plant all around, by the way. This is the pineapple flower, okay, and the fruit develops to the top. Each segment develops from the carpel of one flower, right? So each one of those kind of triangle or diamond-shaped bits on the pineapple is an ovary connected to one flower. So flower, 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 right? So lots of flowers on that inflorescence needed pollinating to fertilize all of those ovaries that then collectively developed as a group into this thing that we classify as one fruit. In contrast, the raspberry only had to pollinate and fertilize one flower to get the same effect from several ovaries, which each one here had one ovule. So there's a gradient of variability.
So if every flower in the pineapple doesn't get fertilized, does it just not grow properly, or?
Every flowering plant that does not get fertilized,
No, so with the pineapple, it's like a bunch of flowers in one organism, and they all have to get individually fertilized. If they don't, is it like one section just doesn't grow, like one chunk?
yes, great question. So yeah, if that doesn't happen, right, there's some kind of pollination problem for some reason, or who knows, maybe something ran through the field and knocked off one side of those flowers, right, then they couldn't be pollinated because they got broken. Then yeah, then you would just have like maybe one side of it where these wouldn't look like the rest. They wouldn't be fleshy and developed. When you cut it open, they would be like, probably they're still green and hard, not sweet and good, and you would just remove that part. Another example of that phenomenon is sweet corn. So sweet corn, I'm going to try to draw it now. So lots of individual kernels on the cob. Right? Stay with me. This is just like one level of what you can see, right? Each kernel is connected, a silk, to the outside. So, if you've ever shucked an ear of corn and you just got a bunch that, it's just still white and there's no like fleshy yellow kernel there to eat. It's just because those individual silks didn't get pollinated. So one after one after one. So I told you that about corn. So there's, right, each one of these has a flower, right? So there's many flowers and there's many ovaries. Then what does that make it? And the cob, right, is a giant receptacle. So it's sort of like the raspberry, but it's what's different is it's many flowers.
So what's the advantage to that? Is there one, or that's just how that plant is?
It's working, right? Well, first of all, anyone else? What's the advantage to it, do you think?
Genetic, diversity.
Diversity, absolutely.
That's
Joshua?
what I was thinking, genetic.
And then, but wouldn't that be the same no matter how many are in there? Andrew?
Fault tolerance.
What tolerance?
Fault tolerance.
Fault tolerance?
Fault.
Like redundancy? Yeah. That's a big one. I think in this particular case, food crops, you also have to look at the ancestor of that corn before it was bred by humans to be like, 1,000 kernels on the ear. There was like 5 to 8 kernels, right? And so there was a lot less redundancy and a lot less genetic variability, which is still, right? 5 is more than 1, so there's something there in terms of variability. But those early ones looked like, you know, 1, 2, 3, 4, 5, and that was basically it. And then, so yeah, then each one of those had a silk, and that's what you got. And people were like, oh, those are pretty edible, you can cook them, but let's get the ones that grow the most. Joshua?
The pollination time, so as it's developing, you might have different, they might be able to receive the pollen from above at different times if the environmental conditions change.
And how's that an advantage?
Because then even if the pollen's not right at the same time as that guy's ready to receive it, then you can space that out for longer.
Right? It broadens the capacity to be fertilized, right, from different plants. If it just so happens, right, that cross-pollinating other corn plant flowers early, flowers late, it's blown in on wind from a distance where conditions are different, yeah, and you've got a broader opportunity for success. Roland?
Does this mean that corn is a fruit?
Corn is fruit.
Thank God. Okay. I think it was a big issue.
Yeah, let me leave you today with the thought that fruit implies that fleshy outside thing with the seed inside. And I gave you a couple examples like banana with the seeds aren't viable, etc., but they've still developed in that way, and that non-viability there is just a result of breeding more than it is of the initial habit of that plant, right? Sans humans. So yes, botanically, fruit is a broad category, and we've sort of narrowed it down to sweet things. Yeah, right? But like tomato, cucumber, corn, peppers, right?
Corn is a grass, right? Like,
Grass
corn is a
is-
grass.
That's right.
Okay.
But still, right, so amount God, but still flowering plant.
Yeah.
Or sorry, not flowering. Still flowering plant. And still develops some way to get the seed out there. And there are other cool examples of grasses that have a impressive seed. Coconut tree, for example. Coconut palm. Right? Amazing. Large seed. Right? It's going to come up in like the next slide or two.
So on that subject, when you're trying to differentiate like a grain from a fruit, there's both grass with corn and grain, is that the pericarp, the way that's made up, that would be the
Yeah, I mean, at a certain extent, we've just made that division where we basically say that grains are the fruit of grasses, right, more or less. And because they're poaceae grasses and they're monocots, the development is all in a single endosperm rather than two cotyledons, and so that makes it look different in there. And then they have, like, in the case of, wheat, for example, there's the germ, right, on top of the embryo part of the seed, but all of them are fruits developing, right, from the result of pollination, fertilization, and then development around that ovary.
So sort of monocot, it just presents differently than what we're used to calling.
Yeah, they were different enough that people came up with specific words for them, but they're still, right, it's still a fruit, or, many fruits, depending on how you
Follows
use it.
that same set of rules as developing.
Absolutely, it's still the reproductive mechanism. It still has a both protective and dispersal role. And if you were to call them all a fruit, you would be correct. Does it?
Yeah, so before you were saying that a fruit is something that has the seeds on the inside, and you said that a strawberry would be considered a vegetable. What is the definition of a vegetable?
So strawberry is interesting because the seeds are technically on the outside, right? But it's on the outside of the receptacle, not on the outside of the ovary. So what you're eating, the big red part of the strawberry, is all receptacle. It's a nice, fleshy, yummy one, but still. And so to your point, there's no like international court where we can say that there's one definition of fruit versus vegetable, but generally the understanding is fruits are developed in this process I'm talking about, pollination, fertilization, right? They have a seed, there's some kind of fleshy pericarp surrounding it, and vegetable is all the other parts of the plant that are not from the flower. So carrot's a root, celery's a stem, what else? Collards are a leaf, absolutely.
Potatoes,
Lettuce is a leaf, kale's a leaf, yep.
Onions.
Leaf.
This is kind of random,
Onions
but
and leaves.
this question reminded me that something that kind of helped my understanding when I took the botany class was when I learned that vegetable is technically a culinary classification, has nothing to do with botany. That helped explain a lot, because I was always like, how is a tomato a fruit, a veggie, and apparently a berry? Because vegetables aren't actually genetically classified.
Yeah,
It's my
right.
favorite little fun.
Thank you, yeah. So what does it really tell us? Is the point, right? We call it one or the other. Like,
Yeah, vegetables what you make it, but fruits are.
You've been
oftentimes
lied to.
connected to that being a non-flower part of the plant. We understand that there's generally more fiber, right? Because you're eating something that wasn't made fleshy before seed dispersal, which I'm going to finish with in just a moment. You're eating a bunch of ground tissue inside instead. But yeah, this is pretty much, we think about that from a cooking, grocery store perspective, and not the tan. We're going to do it in
Brussels sprouts, do they grow?
Brussels sprouts.
Yeah,
Great-
it's
great
a small
question.
step.
Yeah. Brussels sprouts.
I'm like trying to dissect a Brussels sprout in my brain right now.
Great question. Great question. Brussels sprouts. I showed you. Here's the stalk of the Brussels sprouts. Okay? In a blown up version, if you've ever seen a whole stalk, right? There's lots of these, right? Like that. Then you see that actual sprout is here and here and here. So what does that make you think?
It's a bud.
It came from the bud, right? It's young leaves folded on themselves, right? Folded in on themselves. At the node, right? That came from that axillary bud. Yeah, just each time it's there. So we selected for a thing that instead of making a shoot here, right, that when it did something else, it just immediately started to then make these leaves that we go back to our here maybe. No. It's basically this
Yeah, that's what I was just saying.
over and over, and then it gets bigger. So it's still a vegetable. Yeah.
Kind of backtracking, but, so for the corn, is the cob the receptacle, and like, what's the receptacle? Because you said the strawberries outside is the receptacle, and they even have like a little white cone in the inside as well, so I'm curious.
No, that is on the strawberry, that is the receptacle, like the red part.
Yes, that's my point, because like on the raspberry, the white little cone is the receptacle,
Yeah.
so I'm curious
What's the receptacle?
Yeah.
That's the question. I see. The
basically,
receptacle is basically something for all these things
you know.
to stick to. Yeah. Something to hold them there.
Okay, that makes sense then for the strawberries.
Was that like the
So
pistil
it's the
that?
bear
With
of
the
the
strawberry
seeds,
seed
yeah.
outside, oh, it's outside, except for the. So strawberry ain't really no fruit.
So here,
no, the fruit would be just tiny, right? On the receptacle.
right here, this is very large,
It's a seed.
right? And it's something that
I think
all
that's a tiny
these
fleshy layer.
flowers where a little fruit is going to, right, be produced at the base of each one, it's something that they can all connect to. Now, it's less necessary in the case of the simple one flower, one ovary. There's only one thing, right? You don't have to hold multiple parts together.
Okay, so it's just like the carrier of the flowers or seeds, right?
Yeah.
Okay.
It's like you needed to put multiple things together, so you put them all on a plate, right? Instead of just like, you know, have no way to put them all in the same place at the same time. Yeah.
So what part of the, strawberry is the actual fruit? If that's our receptacle, it seems like it's just receptacle of seed then, right?
Yeah.
So it ain't no fruit.
So this is.
I'm just trying to.
It just depends on what stage of development you're looking at, right? There is a time that fruit is there. Each, so back to my aggregate fruit, many ovaries. Each one of those seeds was one of those ovaries. But there it's like,
This tiny.
strawberry has sacrificed the pericarp for big fleshy receptacle as the way to effectively move something around, and then used bright color as an attractant instead of like fleshy yumminess. So there's just a trade-off on how to get things moving that seed around.
So by the time we see it, the fruit pretty much already gone, we just seeing the seed.
Yeah. And yeah, let me make that final point here, and then we'll take a little break. These are all connected to the way that the seeds disperse, and this is closely connected to how the flower structure implies, is it insect pollinated, hummingbird pollinated, wind pollinated? And remember the differences in those pollens between wind or animal pollination, or something that can catch on the wind and get blown around, something that can attract an animal to it. Here, the strategy is, how can we get this thing out in the environment successfully? And you've seen all these in action. Right? Wind dispersal of seed, dandelion's obvious, right? It's got like just like those wind pollinated pollen grains, something to grab the wind. Right? Maples, you know, it's got little wings, so as it falls, it's gonna move around, right? Rather than just fall straight in place. That's gonna disperse the seed, more possibly advantageous environments for germination down the road, more spread. Animal dispersal, we've been talking a lot about this with fruits, right? Something yummy and attractive, the fruit gets eaten, and then it's generally spread in droppings. This gets to, I think, what you were saying about the dog the other day.
Yeah.
Right? Really, they do a better job at seed dispersal than at pollination. So anything that could get caught, like on a burdock or a teasel or something like that, grab onto a fur or a sock.
Well, that's what I'm talking about. Dogs run through all kinds of stuff, and then, you know, you get them at the house, they have all seeds and stuff in their coat.
Yeah, so you would see that something that wants to disperse its seed in that way is gonna do something like this, right? Little mini hooks like Velcro to grab onto something as it passes by. Others are set up to pop. These are a good example. As the pod dries, they bust out. If you've ever seen here, we call it popweed sometimes, or little western bittercress, Cardamine oligosperma. It literally, right, gets to the point where if you touch that pod, it will blast out, you know, like maybe a foot in distance. So it's spreading that way. Explosion or drop and roll, right? Heavy things that can then move a little bit once they hit the ground, in contrast to move a little bit first, and then, you know, it's lightweight in the case of the sycamore or maple samaras, so you end up where you end up.
At the top there, it says that they spread out so they don't have any competition with each other. Is that in terms of competition with pollination?
So now this is seed dispersal, so pollination's already happened, fertilization has already happened. We've created a seed, right? So now this is about successful growth of the germinated seedling.
Oh, it's
Whatever.
competition of
Parent plant. So the seed drop like right below the parent plant, then where does it get its son? The shape.
So that's the concept. So the competition, right, you're never gonna avoid all competition, right? But you do want to spread out. And so it could be from other, right, to spread yourself out from other maple seeds from that same plant. It could be to spread yourself from plants neighboring different species that are going to shade something out more. And so having spreading your seed out over a wide area gives you a chance to effectively do that. You know, there's, on the plant end, they're playing a numbers game. There's no expectation that every seed successfully produces an individual in the next generation, right? But they're playing a percentage game where if I produce 10,000 seeds and I get 10 successes, then, right, the role is complete for reproduction.
So they're basically competing for real estate to be able to run.
Yes. Roland.
Where do aquatic plants fit into this, and like, does seaweed have seeds, like? Oh, okay.
This, excuse me. The seaweed has seeds.
Yeah.
Yeah.
Look that up. I think you will find some of the things that we call seaweed are not in fact plants. And some aquatic plants absolutely do have seeds.
What thing you call seaweed?
I mean, some things are
I mean, like,
Mosses.
what do you mean?
like an algal mass, right? And so if it's an algae and not a plant, then it doesn't reproduce in the same way. There are absolutely seaweeds that are plants, right? And their reproduction is very interesting. Yes, so some produce seeds, some spread in water. I was talking about coconuts. That's one where the seed, right, it's produced on land, but immediately falls into water, oftentimes, and then it floats around, ends up in some other terrestrial site, and then germinate.
Just Googled, and it says that that seaweed or macroalgae, so definitely like the plant ones, use spores, not seeds or.
So what do you think the consequence of that is?
I don't know. I don't know.
So
Furnish.
spores means
Furnish.
not a flower, right? Yeah. So that pollination step is different, which makes a lot of sense. You're basically putting it out into the current of the ocean, and you're just trying to get two things to run into each other. So here, graphed out a little bit, wind, animals, water, top it open, people, right? And what I want you to take away from the seed dispersal is just the connection between the design of the thing, right? cocklebur, sandbird, whatever, sticking to you, you know, milkweed or dandelion flying away on the wind, something that pops, obviously, from the pod, and how that mold connects to that attempt to find good competitive space. And sometimes it's just about total area, and sometimes it's about specific conditions, right? Some things that grow very well as an adult in a denser, less light environment need a lot of light as a young plant, or vice versa. Right? So there's a connection between where something evolved and what those needs are. Like oaks out here in the oak savannas of the Willamette Valley, as it gets colonized with more montane species from either the Cascades or the Coast Range, it gets shadier and shadier. And those oaks are pretty sensitive to developing in shade as a young plant. So it's important that they have, you'll notice if you see an oak grow, it's dispersed, right? They're not all one on the other. They get pretty old and big. And that's because that level of light that they need is only found in certain spots. So the ones that don't get enough don't grow up, and there's not like it's not like a stand of them like gun firs, right? It's just a few individuals spread around. So it's all connected to what that need is. And then of course, the acorn is relatively heavy, doesn't have wings, right? It's just falling. So a big mature oak tree is putting those out over its canopy, but not much wider. You know, squirrels are moving them somewhere, for sure. But the ones on the edge of that drip line are going to be more successful because they've got more light from the side, right? Or the ones on the inside might germinate early season before that whole oak canopy is established again for the next growing season, and then right, they might die in that first year of germination because they're too close to the trunk and there's not enough light penetration. So it's all connected to the individual species need, and of course where they came from initially that set that up, and then adaptation comes into like the threshold between how much light, how much moisture, and can a little added genetic diversity make that range wider.
Could fire be a way that seeds disperse?
Yeah, absolutely. It's less a dispersal mechanism and more related to germination, which is going to be, when I talk about seed germination in just a little bit, that's going to come up. Yeah.
Just because of like, with forest fires, they're, at least like the ones that I've been close to, it gets really windy. Do you think that that would help in like the way of having like?
Well, that might help move some wind dispersed pollen. At the same time, if that temperature is really high, it could also torch it. So it's a double-edged sword there, I think. I think the most effective would still be fire opening up seeds or combs that are really thick and then like need that high temperature to crack it open and make it available. Let's take 5. We come back, we're gonna talk about seed and cut those seeds open. So grab one on your way back in if you haven't already. You're more looking at the flower for what? Oh yeah, definitely. Because where the seed are doesn't tell you a whole lot. This could have 1 or 500 seeds. You know. Same thing here. I mean, there's a combination of ovaries or multiple ovules within an ovary. How many seeds and where they are is highly varied. So this is about just the connection from one flower pollinated to one ovary fertilized, one flower pollinated, any and like to Ryan's question, corn example, same thing. If you only fertilize half of those, if you get like a raspberry that has, you can see it sometimes, right? Yeah.
it is a poor choice, pre-flood, because that would be coffee for $25, okay? Well, then I doubled down, and it turns out Nashville Airport has a cigar lounge inside of it. Got that, and I would sell, let it smell like a cigar lounge. But I did not take advantage of the airport. Yeah. I was the gold artist, and I was like, I was trying to decide, like, what has the installed out of that? I'm sure there's like.
It's definitely interesting on its own right, but like, yeah, I think the CSA part. Yeah, I think the CSA part was the same, yeah.
How much is there?
First 4 weeks, we have not deals mostly because like it's a whole harvest, but now this was the last week. Now we're gonna do more like.
Yeah, it's gonna change quite a bit after today.
Yeah.
It's almost like two different classes.
That's what I feel like it's gonna be. I'm like, here,
They're connected, but one is just all doing and the other one.
it'd be interesting because we did all doing this summer. for the most part, so
Right,
it'd
it's
be
a lot like
a
that.
learning.
And then now there's, the second half will be more context and details like about storage, about marketing, about, you know, there's a field trip to the organic grown company, so we get like the connection to, oh, here's what we're doing, but here's what, you know,
Big scale.
someone else is doing. Not just big, yeah, big scale, but also like, you'll see, we're basically doing what they're doing, they just have more expensive equipment, right? So how do you approximate that, you know, without like a $250,000 square foot warehouse?
Exactly.
Keep it up, you're doing well, see it. This time,
Uh-huh. No, it's-
yeah. just understanding the introduction of grasses.
So that's what we have to do.
Oh, yeah.
We're not, yeah, we're not doing that.
He says no. No. Well, that makes sense too.
These are the same ones from like.
I took those.
Oh, really? Two more today,
Yeah, I was thinking so.
Yeah, and a razor blade.
Apparently, John asked me about that. Walking I was like, so, you just transfer it, what's you? Okay.
5 more hours.
So, that's, right, so, corn's monoecious, but not perfect, right? Same plant, different flowers. The tassels produce male flowers.
Tassels.
And
Mini corns?
And the silks, many are going down. The silks.
I knew.
Part of the problem is flowers and the pollen.
So, we start out with some introduction questions about corn, because I don't
But this is just a curiosity. And then the silks are the female flowers, right, that connect directly to all those ovaries that become.
But these are not
Your mama comes, all
Take
right?
it all the way back.
Well,
Corn irrigation.
You gotta take it all the way back.
dicots are changes, but
And then, this is the one that we.
Who knows? Our monocuts,
I mean-
angiosperms.
they're not, but why do we call angiosperms flowering plants if there's flowers on them?
But that's the male flower.
It's all about the definitions, right? And then even if you do something like,
Monoecious.
go to the internet and be like, are
That's
monocuts,
Yeah, so if
do
you do
monocuts have
it's
flowers?
but, probably get better, growth when it falls on.
Yes, monocuts do have flowers, Andrew. See? Let's go back. So, in our first week's evolution,
Big, broadleaf, like.
right, we had all the plants generally. We had like the ferns and mosses as the spore producers. Right? So ferns and mosses. And then from there, we went to gymnosperms versus angiosperms. And from there, we went to mono and dicot. So they do have
I think you got gymnast.
I got that all wrong.
Mixed up with. Kind of some bad kind.
It's harder because they are so different, right? You've driven past like a field of wheat in the valley or out on 84 East or whatever. Those are flowers, right? What looks like on barley or wheat as just this sort of thing repeated, right? That's all the flowers. Like wheat stalk. Let's get through seeds. So, get your seeds if you haven't already. You'll notice one is a bean. To the point we're just discussing, one is a bean, dicot, and one is a corn kernel, monocot. So, we're going to see something different about them as we discuss this stuff. So there's a reason to have both and compare. So some of these terms will be familiar from the last 2 days of talking about this, but I want to just zero in on it along with the looking at them. So if you have a hand lens, today's the day. And then I've been trying to zoom in as much as I can with the doc cam too. on the cutover ones. So a seed is a fertilized ovule, well, what we've mentioned, and to Christopher's question earlier, surrounded by a seed coat, just like everything else, right? The epidermis on the leaf, the epidermis on the root, everything has some kind of protection. The seed coat is that for the seed. Holds it all in, keeps it from losing all its water and drying out. And also in cases of food supply, which is either cotyledons on the dicots or endosperm in the monocots. In both cases, it is the food supply for that seedling plant. And so here's, when we cut open our bean now, here's what we're going to see. You see a thing tucked in right at that spot where it looks like there's kind of an oval spot on the bean, like a belly button almost. And that's going to be where the radicle, the first root, is. And that's the first thing that will come out of the seed coat at germination. You can notice in the bean too, there's a little leaf there. Call that the epicotyl, the part that's going to be the shoots and leaves of the plant. That be above the cotyledon, and hypo below, that's going to be the stem and the root. And this is kind of the dividing point there. So all of this is cotyledon, and as those develop and flip out, then they look like the things that you've seen before on that seedling plant. Surrounded by the seed coat, and that's our model for seed about to germinate. Again, here's another way to look at it. Remember, the food reserves are the cotyledons. This is just the way that you can see, okay, there's two of them, right, or the dicot, and then baby root, right, future stem and leaves of the epicotyl. And in contrast, in the monocot, again, we call that food storage area. The endosperm still has seed coat around it, still has baby root, which is still the first thing to come out of the seed coat. So why in both cases does that radical baby root come out first?
Is it to hold it in place?
Hold it in
Or
place.
to get water?
To get water, right? What's job number one of successful living plant? That's why we started with roots. Get water, absolutely. Can't do any of the other stuff until that objective is achieved. So get out there, right? Differentiate, make a root hair, absorb water. That's our goal, monocot or dicot. So then it's important to recognize that these things are well positioned to survive in the environment post-seed dispersal, to wait for a good condition, to wait for when the competition level is right, whether that be enough light, right? So that means it's not too densely packed with other plants around it. And we call those factors seed dormancy. And I'm going to talk about breaking dormancy is how we get out of it to the point that we can grow the next plant. So, in the beginning, the seed coat keeps the seed dry and safe. And then internally, there's a series of metabolic signals that are keeping this seedling seed plant from germinating. And the balance of those is keeping it in place. And then something switches the signals, so it says, time to go, time to start growing. And there's a series of factors that impact that are the things involved in breaking seed dormancy. So you'll notice, radicle is the first thing out. Baby root, root cap, and root meristem here. Not differentiated, not differentiated, not differentiated. Now root hairs, right? Lots and lots of root hairs. Using all of that cotyledon energy to produce those root hairs. Can't do photosynthesis yet. It doesn't have any leaves. So that's the only thing that's available for growth at this point. Is the cotyledon food source, or the endosperm food source, to make sure that root is produced. So what are the factors that get us to that, breaking that dormancy? Number one is imbibition, or water. Right? That's why before I brought these bowls of seeds to class, I soaked them all morning so that we could see some of that development. It would soften up the seed coat. It'd be easier to cut, and when you peel it apart, you can see the individual pieces more easily. So water is your number one way to break seed dormancy. Temperature plays a role. Different species have different range for that. Some need treatment at a cool temperature. You might call that stratification. Some maybe you've played with, you need to put them in the fridge for a couple weeks, right? It needs to be around 40 degrees for a while. Some might need to be stratified at a warmer temperature to induce germination. But water, temperature, scarification is another one. That's physical abrasion. Right? It's in the natural environment, that might be actually being in the soil, bumping into microorganism or gravelly sand or something and actually getting abraded. Another common one is going through animal digestive system, stomach acids, basically stripping away the outside of the seed coat, making it available then to take up water. Oh, that's scarification, like literally scarring the outside. If you know you're trying to propagate something that requires scarification, sandpaper is a common way to do it. You might sit there and go like this on it. You might use a acid solution to recreate that animal stomach effect. When I was in school, we had to scarify lots of seeds for testing different things, how quickly they would germinate. We had a little box lined with sandpaper and a vacuum. And so we would just run it, and it would just spin all the seed around, hitting all the sides of sandpaper over and over and over. And we do that for like 2 minutes, and then that was the scarification treatment. So water is a temp, physical contact with scarification, in some cases red light, stratification I mentioned, specific temperature treatment, fire, getting to Zach's question, chemical inhibition, and the radical is the first thing to emerge. So you've got the seed, it could be in the right environment, but the conditions might not be right. The conditions might be right, but it's not in the right environment. Both of those things are going to impact how readily it germinates, how readily dormancy can be broken. So when we get rid of dormancy, when we break it, then it's no longer dormancy, and now that seedling can start to grow, which we call germination. I just wanted to point out, sometimes you hear germination and emergence used as the same thing. Germination is the plant's actual waking up process as a result of one or more of those factors from the previous slide. Right? It's the actual biological breaking the seed coat, radical coming out. Emergence is when we see it over the surface of the soil or the pot, media, whatever it is. They've classified the types of emergence into 2 types, epigeal, epi again above, or hypogeal. And that just tells you about where the plant parts are when that happens. So in epigeal germination, so this is your cotyledon. It takes place here, below, and in hypogeal, right, that hypocotyl is all below, and we just get the leaf. So don't worry about that distinction too much. Just want to mention that, right, there's 2 different ways. And basically, different plants choose one or the other. There's a slight advantage to having the hypocotyl be above. You can protect what's going to be the baby leaf there underground for a minute until it turns and flips over, versus in epigeal, when it immediately comes up, right, you see everything right away. There's a little bit more risk. Something could immediately come and eat that little baby first leaf right away. On the other hand, right, there's no barrier to growth, so it's faster takeoff. Here you still have to break the soil and need the energy to pull that up and out. So the beans work like A in performs like the peas do here in Armstrong. So safety at germination, above ground or under. Once dormancy is broken, variety of factors that impact it. And once dormancy is broken, now we've germinated. For the germinated seedling, I just wanted to show the importance of temperature. Right? So beyond that stratification that might say now dormancy is broken and you can grow, when you're actually growing as now a seedling, right, there's a range of temps. So here's just a generic layout. You can see there's some peak temperature, right, and a gradient. There's too hot for growth, there's too cold, and there's effective growth at some range in between. So knowing what that range is for the thing you're trying to grow is really helpful. Generally, the germination temp, the best temperature to get germination the fastest, tends to be pretty high, usually 75 to 85 degrees, and then the best growing temp tends to be lower than that. So again, that's a general rule, not consistent for every species or anything, but when you're trying to force something to germinate quickly, you want to keep it wet, you want to keep it warm, and then if you want to continue that seedling growing quickly, it generally means changing those conditions. You now want it to be moist, but not wet all the time to avoid disease, and you want it in the range of optimal growth, not just optimal germination temp. So keep those two things separate, germination temp and growth temp. So this one's from the homework. You recognize it if you've looked at it already. Here is a germinated seedling over a period of days. Right. Dormancy broken, radical comes out. This is a measure of how much dry mass the seed has. So that means in science we think about it as if you cooked off all the water, take all the water out, what's the weight of the dry portion of the plant? So here it's fairly high for that seed, then it drops down, drops down, drops down, drops down, and it doesn't start to rise up again until here, and it doesn't reach more than what the seed weighed until well over here. So my question is, what's going on in this section?
Burning through its energy.
Burning through its energy.
And then photosynthesizing to get
Say the last part again?
photosynthesizing like why?
Yes. So yeah, first, using up that cotyledon or endosperm energy. That's right. And so it's literally being consumed, thus there's less of it. So total weight's down. And then, now that there's cotyledon plus true leaves, photosynthesis can happen, food production, then we see actually more mass because the plant's growing. So let's take a look. You got your razor and your seeds. By the way, the empty small hole is for the razors at the end. The bean seed is a classic example and relatively easy to slice in half. See, I just popped it through that belly button, called the helum, and then it looks like so remember, at that middle section, we said that's where the radical is. So this is radical right flower anatomy, pardon me for running out of time, we had a lot of good questions there that I wanted to address. So my idea here was that you would go out either in the greenhouse, in our gardens immediately around, find a flower, and then answer the questions according to it. So I realize we're just about at time, and if you don't have additional time for that, you got another class, by all means, do it somewhere else, you know, outside your house or at a park or whatever, or if need be, you just choose a photo online and answer the questions accordingly and include that.
Is there a way to look at a seed and know if it's gonna germinate on the below ground?
Just from observing like this, it would be very hard to say. So if you cut the bean and the pea open, they look virtually the same, except for one's more circular and one's a little bit oval shaped. So yeah, tough to say. Mostly you would either observe or look it up. Sometimes you can intuit it working backwards from like the environment that something is in. Most monocots, most grasses tend to be the example that's straight up and out fast, that risks getting chewed on, but they're set up to respond quickly with more growth. So you can kind of generalize that one, but no surefire way. Any questions? So the flower anatomy is just meant to be practice at those things, becoming better acquainted with the terms and where they are. That's something you'll be expected to identify on the midterm, so it's an opportunity to practice with it. And the seed observation too is another opportunity to get comfortable with the names of the parts, how they work, and seeing that, oh yeah, peel that seed coat off, then it's just this. Right? So the goal of germination is to get that seed coat pliable enough that radicle can push its way out. So without imbibition, water coupled with either temperature or scarification, something else, there's not enough force from the radical to push its way out. It needs to soften up that seed coat with water in order to make that possible. Secondarily, I think there's an advantage there because once it's out, then it needs to absorb water to survive. So by water being the main factor in growing dormancy, it also guarantees that there's water present for growth once that root is out there actively trying to grow. What's that?
So then it knows if you are.
Yeah, then it's right, like, there's a much better chance for success. It's like, well, the first thing I need to survive is water. Now, I've gotten the signal that there's enough water, so when I put my root out there, there's probably some left. Of course, if humans like, right, now I took these out of the bowl of water, if I let them there, then they'll still dry out and die, right? But,
We want to plant.
yeah.
Okay.
They only get one shot, so they have to make sure they.
That's, yeah, you wanna get the best chance possible for success.